like to introduce Edmund Neo. Edmund is ProImmune's immunology sales specialist who's located in Singapore, and he represents our company in the Asia Pacific region. Edmund graduated from the National University of Singapore and worked for Abbott Labs before joining ProImmune in 2019. And he's really played a pivotal role in the team supporting customers across the region, and really very pleased to have Edmund on the team, and he's going to be talking to us about epitope identification and clinical immune monitoring tools. So over to you, Edmund. I'm just going to get everybody to mute first of all, and let's do that first.
Hopefully not for me.
Yeah, Edmund, if you can unmute, and then we'll carry on.
Yeah, unmute myself already. Thank you very much, Jeremy, for the great introduction. And I'm glad to have this opportunity to share more about ProImmune as well as our contribution in the area of SARS-CoV-2. Yeah, so you might be wondering who are ProImmune for some of you all. And that is a great question. So in short, if you are looking to characterize immune responses, ProImmune can be a strategic partner to fully characterize both the wanted and unwanted immune responses using our state-of-the-art proprietary technologies that we can offer as a product or service. In more than 20 years of experience, we are the world leader in providing products and services for preclinical and clinical immunology research. We have been serving almost all the expert immunology academic groups, large biotech, and the top 10 pharmaceutical companies. Here I've listed some of the publicly disclosed institutes and organizations that we have worked with before, ranging from monitoring T-cell responses to screening immunogenicity in biotherapeutics program. At the very bottom, here's a picture of the Magdalen Center building where ProImmune's HQ and labs operations is located, and that is where your host Jeremy is located too. Hopefully, soon traveling can go back to normal after the pandemic, and we welcome you to drop by our office to give us a visit if you have the chance to visit Oxford one day. We also have sales support team located in US and myself in Singapore too. So before introducing our technologies, I would like to first explain about the role of the peptide antigen-specific T-cell responses and the MHC molecules using this simplified cartoon. Some of you might be already familiar with this, but this is still important to explain so that we can better appreciate our MHC multimer technologies. In this slide, we have the MHC molecules classified into the MHC class 1 on the left and class 2 on the right. Here we have the antigen-presenting cells, also known as APC, which can undergo proteasomal cleavage pathway expressing MHC class 1 and endosomal cleavage pathway expressing MHC class 2, presenting the peptide antigen on the surface of the APC, represented by the orange and the red diamond respectively. We then have the T cells recognizing the peptide antigen loaded on the MHC molecules through the T cell receptor. The antigen-specific CD8 T cell recognizes the peptide antigen loaded on MHC class 1, while the antigen-specific CD4 T cells recognizes the peptide antigen loaded on the MHC class 2. These peptide antigens that are presented and recognized by our T cells are also known as the T cell epitope. Now that we have been introduced about the peptide antigen-specific T-cell responses, the next question is, what attributes does a technology need to possess for immune monitoring? So it needs to be a robust and rapid technique to evaluate specificity, function, and frequency of immune responses so that data generated can be used to determine correlation between treatment and clinical outcome, thus assessing the efficacy of vaccines and immunotherapies. So this is where in ProImmune, we are the leading expert in providing reliable MHC multimer technologies to enumerate antigen-specific T cells by using our Pro5 MHC class 1 pentamers and Pro-T2 MHC class 2 tetramers. Apart from that, we can provide peptide synthesis for high-quality pink peptide products to be used for your projects, such as performing ELISpot assays for T-cell immune monitoring programs. Now, if I may bring your attention to our Pro5 MHC class 1 pentamers to enumerate antigen-specific CD8 T cells, usually by flow cytometry. On the right side, you can see the design of our pentamers, which consists of 5-MHC peptide complexes that are pentamerized through the COI domain. And at the very bottom of the molecule, each pentamer comprises up to 5 fluorotext or biotext for bright and efficient labeling. This pentamer technology is unparalleled, cited by experts around the world, which you have seen in some of the earlier talks by our expert speakers today. If you face any difficulty, we have an expert technical support team of immunologists that is capable of providing rapid professional technical advice. We have more than 650 catalog specificities that include SARS-CoV-2, cancer, autoimmunity, hepatitis, dengue, tuberculosis, HIV, EBV, CMV, and you may find many other areas on our Pentamer catalog webpage. If you do not find the specificities of your interest, we can provide a custom pentamer synthesis for the mouse, human, and non-human primate alleles. So here is a case study by Professor Tao Tong from the University of Oxford, who have joined us earlier in the first panel discussion just a while ago. She is one of the first to publish a paper describing T-cell epitopes for SARS-CoV-2. With the close relationship that she has with ProImmune, we were able to work closely together that enables her to rapidly assess large quantities of reliable custom pentamers. This unlocked the ability to perform in-depth analysis of SARS-CoV-2 T-cell responses in COVID-19 infected patients and vaccine-treated population. At the bottom right, you may find 6 pentamer staining data, which earlier Yen-Chun Peng has shown, with the respective HLA restriction labeled on top of each plot. If I may explain about the data interpretation, the x-axis of each plot is the pentamer and the y-axis is the CD8 T cells. The cell population are located on the top right-hand corner, which is both pentamer positive and CD8 positive. Thus, this population is our antigen-specific CD8 T cells that we are interested to look into. If you're interested to find out more about her work, there's a great talk on our YouTube channel given by Professor Tao during our Mastering Immunity last year, and even by Yen-Chun Peng earlier during her talk. So moving on, with the paper describing some of those T-cell epitopes, Professor Yu-Cho Shin from Korea Advanced Institute of Science and Technology, who have spoken earlier as well, has successfully stained positively using these 2 pentamer specificities to the SARS-CoV-2 spike protein that are AO201 restricted, staining on different infection profiles of COVID-19 infected patients in his study. These infection profiles include acute phase, early convalescence, late convalescence, non-severe, and severe. In-depth analysis of multiple biomarkers gated on pentamer positive cells with a total of 16 biomarkers that include activation markers, exhaustion markers, cytokines, and more. With this data, this paper has shown that the CD8 T cells memory formation may have differences between mild versus severe recovered patients, which indicates that the severity of infection induced a different immune response. Also, it has shown that CD8 memory T cells formed in natural infection may have suboptimal responses to reinfection. Therefore, this indicates that vaccination may be required to develop effective memory T cells. Lastly, which the title of the paper speaks for itself, PD-1 expressing SARS-CoV-2 specific CD8 T cells are not exhausted, but functional in patients with COVID-19. Therefore, this has showcased the importance and the reliability of using our pentamers for immune monitoring program. So moving on, this is our Pro-T2 MHC class 2 tetramers used to enumerate antigen-specific CD4 T cells. We have more than 180 catalog specificities that includes cancer, autoimmunity, allergy, influenza, hepatitis, tuberculosis, HIV, EBV, CMV, and even therapeutic antibodies, and many other areas that you can find on our website as well. If you are not able to find the specificity of your interest, similarly, we can offer a custom tetramer synthesis for the human and mouse alleles. So to show a little bit of the data, on the bottom left, we have staining data shared by George Lauer from the Massachusetts General Hospital and have kindly provided this staining data using the HCV specific tetramers of different specificities for the DR4 and DR1 HLA type donors respectively. More data on the bottom right, we have the staining data shared by Ellie Barnes from the University of Oxford using our HCV tetramers on DRB 1501 vaccinated donors. The top 2 panels are the staining data at 12 and 22 days culture respectively, and the bottom data is using a HLA mismatch vaccinated donor as a negative control. So hopefully somewhere down the road, we can start seeing some of the tetramers staining for the CD4, antigen-specific CD4 T cells soon. So apart from our MHC multimer technologies, we are capable of providing high quality peptides for your immune monitoring program. This is the AstraZeneca Oxford vaccine trial monitoring vaccinated participant SARS-CoV-2 specific T-cell responses using Ting peptides products. Later on, you can hear more directly from the co-author of this paper, Professor Katie Ewer, in a while. So a total of 253 15-mer peptides overlapped by 10 amino acids spanning the entire vaccine insert was synthesized by ProImmune. So for the Oxford group that is able to work closely with ProImmune, the group was able to rapidly obtain large quantities of reliable custom peptides for their clinical trials, and importantly, on time. These peptides were then used in their ex vivo interferon gamma ELISpot assays to enumerate antigen-specific T-cell responses shown in the data below. So today, all I have time is only for monitoring wanted immune responses. And we have other tools that we can offer as a service listed here such as the antigen presentation assay, T-cell assay, cytokine release assay, and many others that is listed in this slide. So looking in the area of unwanted immune responses for biotherapeutics programs such as monoclonal antibodies, bispecific, recombinant protein therapeutics, therapeutic peptides, and many other areas. So if you are interested to find out more, please feel free to contact us, and we are happy to schedule another time after this conference, for example, to have a chat with you for us to explore further on the capabilities that ProImmune is able to assist in your project. So in summary, for a successful epitope discovery and immune monitoring program, this requires detailed and efficient characterization of antigen-specific T-cell epitopes for immune monitoring program. In order to achieve this, it is important to have access to a wide range of tools such as the MHC multimer technologies and peptides, or even more other kind of assays that you might need help with that ProImmune can offer as a service to enable appropriate and rapid decision making. ProImmune is the world leader in providing MHC multimers, both class 1 and 2, for more than 20 years, as evidenced by our customers' extensive publication record. We have the breadth of experience and integrated platforms that allows you to save time, money, and most importantly, reduce the overall risk of your project. So I will end off here by leaving ProImmune's contact details. And if you have any questions, please feel free to drop us an email or drop us a phone call, and we are more than happy to have a chat with you to see how we can assist in your project. Thank you for your time.
Great, thank you very much, Edmund. Whistle-stop tour there of all the different things that ProImmune can help you with. As Edmund said, if you have any questions, please just either drop them a message in the chat and we can get back in touch with you and help you with any questions that you've got. So again, thank you very much, Edmund.
Yeah, absolutely.
Perfect, thank you.